lecture 2. Okay, so let's start. So the first thing is bacterial cell or colony concept. Okay, so here this is a petri plate. This is a petri plate where bacterial colonies are grows. So each of these dots are one colony. Okay, so what is a bacterial colony? So suppose this is a locality. This is a locality, and within a locality many houses are there. So this is a house, this is another house, this is another house, this is another house, this is another house. So in different house, in all these different houses, in a single house, the members are there, all are called, they belong to same family. Okay, so in a single house all the members are their family member because they belong to same family. In the same way, for bacterial colonies, in a single colony, if we zoom a single colony, we can see in a single colony many microorganisms are there, many bacteria or bacterial cells are there in a single colony, like 10 millions or 10 to the power 7 individual cells are present in a single colony, and all these single colonies are came from a same species. They are belong to the same species because they are present in a same family, because they are produced or generated from a single bacterial cell. Suppose in a petri plate one bacterial cell is present was present, and then it divide to two bacterial cell, and then this bacterial cell divide to another two different cell, this bacterial cell divide to another two different cells. So from this single bacteria in a colony, all these bacterial cells produce different many bacterial cells in a single colony, and all they come from a single ancestor. Okay, so they are all belong to the same species. So within a bacterial colony, same same species of microorganisms are there. So within a colony many microorganisms are there, and all the microorganisms or all the bacteria are within a belong to a same species. Okay, so this is the concept of bacterial colony and bacterial cell. And for our any experiment, when we experiment or when we count the bacterial cell for our any experiment, that time we do not use the term bacterial cell, the count of bacterial cell. We use the term bacterial colony because we cannot count the bacterial cell because it is very minute, very small, and they're present in huge amounts. So that's why we can't count the bacterial cell. We can count the bacterial colony, and we called it colony forming unit. So we called it colony forming unit. Okay, CFU. We denote it as a colony forming unit. Okay, so